Good afternoon and welcome to Classic Sci-Fi Movie Reviews. I'm your host, Reginald Pumpernickel III. Today we'll be discussing the much beloved classic, The Fifth Element. And with me in the studio is 2022 Beckman Young investigator Dr. Dylan Domaille, an expert on The Fifth Element. Doctor, welcome. Thank you. Now my first question, Doctor, is, to you, what is the fifth element? Well, as a chemist, to me, the fifth element is boron. In my lab, we develop molecular tools that allow us to track signaling compounds that are produced by cells. And like the theatrical fifth element, boron serves a critical role in the tools that we developed. It confers very high specificity for the compounds we're interested in. I'm sorry, did you say molecular tools? Actually, yes, and boron is the atom that is activated by the compounds we're interested in. So my lab is interested in studying oxidants that are produced by cells. These are necessary for healthy cell signaling, for immune function, but if too much are produced, or if they're produced at the wrong time, or if the wrong type are produced, it can lead to things like chronic inflammation. So there are a lot of different oxidants that are produced by cells, including things you'll see at the pharmacy, like hydrogen peroxide or bleach. But we're interested in one called peroxynitrite. And it's historically been very challenging to study peroxynitrite when there are all these other oxidizing molecules around. So the question is, how do you design a molecular tool that is very specific for peroxynitrite? And what we found is that by using a diazoborane, which has a boron atom locked into a ring structure, this kind of chemical motif only reacts with peroxynitrite. It reacts very slowly with all the other oxidants. And so we couple that reaction to the generation of a fluorescent molecule, and this allows us to see when and where peroxynitrite is being produced.
My name is Anna. I'm a graduate student in the Domaille lab. My work is focusing on detecting peroxynitrite in living cells, and to accomplish this, I am making chemical tools that change color upon reaction with peroxynitrite. This is one of the compounds that we developed. This is a ratiometric compound. In solution, it emits a blue fluorescence, and this is our reference signal. When it reacts with peroxynitrite, it undergoes an irreversible chemical transformation, which causes its fluorescence to change to green. We can use this color-changing property in cells to get an idea of when and where peroxynitrite is generated.
Absolutely fascinating. Now, can you use these diazoboranes for anything other than detecting peroxynitrite? Absolutely. And the utility of the diazoborane doesn't just stop at that single compound that Anna showed. We have an entire tool set of peroxynitrite detecting probes that rely on this diazoborane motif. We have one compound that starts dark and it becomes green fluorescent when it detects peroxynitrite. Anna showed the color changing one, and we have another compound that will change color depending on which oxidant is present. So if it's hydrogen peroxide, it emits blue fluorescence. If it's peroxynitrite, it emits green fluorescence. But we've also used this chemistry to cross-link hydrogel networks. So think of something like a soft contact lens. That's a hydrogel. It's a water-swollen polymer network. And it's very important if you're using these kind of polymers in cell culture that the hydrogel forms very quickly. Ideally, you want it to be able to encapsulate the cell. And so for that to happen, we need very fast kinetics. So we use this very related motif that relies again on a boron atom to catalyze very fast bond-forming processes.
Hello, my name is Luke, and I'm working on redox-responsive hydrogels in the group that are able to change mechanical properties in response to oxidizing agents produced by cells, which will help us better explore the relationship between redox biology and mechanobiology. So in this dish I have a 4-arm amino-oxy PEG, and I will add 4-arm formoboronic acid PEG, which will form oxime crosslinks, the formation of which are catalyzed by the proximal boronic acid, allowing for rapid gel formation at physiological pHs. So I'll add that now. So we can see that the gel has formed.
So, who do you think are the unsung heroes in The Fifth Element? You know, I'm actually going to say the scientists who managed to clone Lilu from a severed hand. I mean, think about, without that advancement, then Lilu is never cloned, which means that all the downstream positive ramifications that resulted from that don't happen. And that's the way that we approach science in my lab, where we develop new chemical tools to open up the experimental landscape for scientists to ask questions that previously they weren't able to ask. And it's very much in line with the way that Dr. Arnold Beckman thought about science. The development of ways to measure the UV vis absorption of a molecule or measure oxygen content in an atmosphere. These were enabling technologies that had massive ramifications and repercussions, positive ramifications in history. Fascinating. Thank you for your time, Doctor. And thank you for joining us here on Classic Sci-Fi Movie Reviews. Join me next time, where I'll be interviewing a stop codon as we discuss Terminator 2. Good night.